Recording?
Okay, what's going to be due is obviously you need to know pretty much everything about mitosis and meiosis. So the one assignment that I gave you guys is you need to put together a bunch of flashcards. So we're actually going to start with mitosis. And what mitosis is about is it's actually about nuclear division, which means we are dividing a nucleus. So big picture, we're going to start with one nuclei. And remember, this is inside a cell, but we're just going to talk about the nucleus. And then that is going to turn into two nuclei. But the thing about mitosis that's different is that whatever genetic information is in this cell will end up in both of these cells, which we call daughter cells. So inside here, as you all know, our cells are made up of, the DNA in our cells is contributed one set from mom and one set from dad. So here I'll do a large chromosome from dad and a small chromosome from dad. We'll call that chromosome number 1 and this chromosome number 2. You always number chromosomes from largest to smallest. So here's the ones that mom gave. Now in the center of each of these, we put what's called a centromere. And the centromere is going to hold them together. Then what happens is after we have mitosis, the result is going to be two daughter cells with nuclei that are exactly the same as this. So then again, in each of these, we'll have a large chromosome from dad, a small chromosome from dad, and a large chromosome from mom, and a small chromosome from mom. And again, each of these is going to be held together in the center by a centromere. So what we are going to talk about is we are going to talk about the process that occurs from here to here. And that's something you're going to be held accountable for. So let's start out with something to give you background knowledge so you can understand. What we're going to do is we're going to look at the chromosomes or the DNA. You will see DNA in 3 different forms. One form is much like this. It's so long and it's so thin and so skinny we cannot see it with a light microscope, so we have to use an electron microscope. And this is DNA. This was actually the largest discovery of the 20th century because this holds the recipe that makes each and every living thing what it is. It's the directions for how it can be alive. So when you see DNA in this form, it is called chromatin. So that would be chromatin. But if you had to work with DNA and it was like this, it would be very easy to get tangled and stuff. So to overcome that through millions and millions and millions of years of evolution, our cells learned how to package it up neatly. So what we always do with our chromatin is we will take it and essentially have a spool, and we can wind it around a spool. And that happens with little proteins, and the proteins that we wind it around it are called histones. So then when we wind it up, our chromosomes, or chromatin, looks like from this to something like that. That would be one strand of DNA, and this would be a copy of it. So this would be called a chromosome. And this thing in the center here is the centromere. That's an R there. That is the centromere. Now the centromere is important because not only can it hold the chromosomes together, it's used to move the chromosome throughout the nucleus as needed. So I like to say if I want the chromosome over here, I go hold it on the centromere and say, hey centro, come here. So centromere is what is in the middle. Now, this chromosome, as you see, actually is two distinct pieces of DNA, like two different threads of the same chromosome, but it's a copy. So it's like you have a backup copy. So if we're going to divide, we can always move that, and we'll know that each new nuclei will have a new chromosome. So each of these, what we're going to call it, is a chromatid. And then together, these would be sister chromatid. And this is important that you know what these things are called, because when you get a problem, if you don't know what we're talking about, you will not be able to solve the problem. So that's your background information that you need. Now let's get back to mitosis, or let's talk about cell division. Cell division is a big cycle, a cycle meaning that it flows around and around. So we're going to start here with one cell, and the very first cycle actually consumes the majority of the cell's life. And this big cycle here is called interphase. So that is called interphase. Now interphase has three different components to it. The first part of interphase, I'm just going to estimate time, is going to be G1. Now in G1, a cell is being a cell. G stands for growth. So the cell's doing what it's doing. It's eating, it's breathing, it's living, it's doing its job. It's making proteins from its DNA that it needs that actually help all living things function. This could be a single cell within a larger organism that could be in any part of the organism. So all cells have different types of jobs. But this is our normal growth stage. And this is a cell just doing its job. Okay. Well, all of a sudden, when cells grow, as we remember, they can only grow but so large because their surface area to volume ratio, the surface area does not grow as quickly as the volume does, so eventually it cannot eat as much as it needs to survive. So right here we're going to have a G1, and I go check point. So that is the G1 checkpoint, which happens right here at the end of G1. At that G1 checkpoint, the cell makes a decision. Am I going to divide, or am I going to stay exactly like I am and just not move from anything and stay there? Well, if it decides to stay and not do anything else, the cell kind of leaves this cycle and it goes into something called G0. And then that cell will just continue to do what it needs to do until it dies. Now, if it goes past here and it's going to divide, we then go into S phase. Now, S phase is synthesis phase. Now, at this point, the chromosome is in this chromatin form, so you can't see it. So I'm going to actually use a chromatid instead. During S phase, it just has one, but it knows it's going to have to divide, so it makes a copy. Now, you can't see it yet because it is like this chromatin, but it is not wound around the histones. So as you can see right now, we have an extra set of DNA, so we can copy. Then what we're going to do is, okay, that's all well and good. So now what we're going to do is DNA is copied. I want to make sure I have, can you hand me that book back there? I want to make sure I don't miss something for you guys. Then as we come over here, what we are going to do is we are going to enter our G2 phase. Give me one moment. Now, in our G2 phase, this is when the cell is actually going to get ready, the nucleus, to divide. But in order to divide, let's take a look at a cell for a second. So I'll move this aside. Here's a cell. So far inside the nucleus, you wouldn't be able to see this. We've taken our strands of DNA that we've had. So we have these little strands. And then what we've done is we've made a copy of each of these. And I'm going to draw in the nucleolus right here because that can make our ribosomes. And I'm going to put like a mitochondria right here. And maybe I'll put a little bit of endoplasmic reticulum, and maybe I'll make it rough and put some ribosomes on, and maybe have some free ribosomes out here floating around in the cytoplasm. Okay, now this is kind of where the cell is. It has already done this. It's been through G1, it's grown, but now it's going to get ready and it wants to make 2 new cells. Now this is an extreme example, so it's not true, but here we only have 1 mitochondria. If you're going to make 2 new cells, can you split 1 mitochondria in half?
No.
No. So during G2, the cell is going to make more mitochondria so both sides can have it. Maybe it's going to have to make a little bit more plasma membrane. So it's going to maybe get more phospholipids. Maybe put a bunch of vacuoles in here because we know there's different vacuoles and things can touch things and move stuff around. So in G2, what the cell is doing is it's getting ready for the big move. It's getting ready to replicate. Well then, at the end of G2, there's one more checkpoint. Okay, this checkpoint is like what parents do before they ever leave the house. They say, does anybody need to go to the bathroom or anything else? So here they're just saying, is the cell ready to replicate? So they want to check, do they have all enough stuff? Well now, let's say, okay, if it is ready, now what we're going to go into is our mitosis. So now let's go to mitosis. Mitosis has several different steps. Now I remember mitosis is mat. Can everybody say that? Mat.
Mat.
Okay. This is interphase, which we just covered the majority of. This is prophase. This is metaphase. This is anaphase. And this is telophase. So those are the stages that we are going to look at. So let's start with our prophase. Okay. During prophase, this is right at the very end of interphase. Again, I'm going to draw our initial cell. There's the cell membrane. I'm going to draw an extremely large nucleus just so we can see what's going on. I'm going to put a nucleolus in here. And then the first thing you're going to notice in prophase is this is how it would look at the end of interphase. You really can't see much of anything. Then all of a sudden, suddenly, the chromosomes are going to condense. That means the chromatin is wrapped up around the histones. It gets shorter. It's all wound up, and poof, suddenly you can see it. So it would be like out of nothingness, this would appear. So here in the cell, we know we already have copies. There's a large one from Dad. Made that too big. A smaller one from Mom, so I'll make that one a little bit larger. A large one from Mom. And a small one from Mom. So that's what it's going to look like. Now a couple different things are going to happen. So the first thing that we noticed in prophase is here our chromosomes condensed. Another thing that's going to happen is you're going to start seeing these 2 little things here, up here. And between them you're going to see all these little fibers. These are called centrioles. And these fibers are called spindle fibers. So this is what you'd want to really have on the front of your flashcard. But now the purpose of these spindle fibers is they're like tow trucks. So these are going to connect to the different chromosomes and pull them from side to side. Okay, now so everybody should be not texting, Dylan and stuff, and looking here. Okay, yeah, thank you. Okay, so this is what I put on for your parents. You had Dylan, stop texting, because when you fail the test, I'll just refer your mom, send her this link. Okay, so this is what we have now, but I like to refer to these as tow trucks, and these spindle fibers, mind you, which are microtubules, which we have already studied, they need to attach to this chromosome, but can't you see that the nucleus is in the way? Dylan.